What allows us to do the things we love the most?
In terms of movement, we rely on cells called motor neurons. Upper motor neurons send info from the brain to the spinal cord, while lower motor neurons send info out to our muscles, allowing them to contract and us to move. A critical gene involved is the SMN1 gene, short for survival motor neuron 1. This gene codes for SMN protein, one of multiple proteins in the SMN complex. As the name suggests, SMN protein is crucial for the survival of motor neurons, and insufficient levels can lead to cell death. This is what happens in people living with spinal muscular atrophy, a disease characterized by dysfunction of lower motor neurons, leading to muscle weakness and wasting. Around 95% of people with SMA have a deletion in both copies of the SMN1 gene. Although there exists a backup gene called SMN2, only 10% of the protein product is fully functional. People who have more copies of SMN2 can experience less severe symptoms of SMA. However, overall, there is still not enough SMN protein produced, leading to difficulties performing basic movements. This is particularly devastating in infantile onset SMA, since neurons cannot regenerate and damage can be permanent. So what treatment options are out there?
Zolgensma. Zolgensma is a one-time gene therapy administered during a single 1-hour intravenous infusion to children under 2 years of age with SMA. It introduces a new copy of the SMN1 gene packaged inside a viral vector to motor neurons. Adeno-associated virus 9, or AAV9 vector, transports this gene into the motor neuron. The viral DNA has been removed so that the vector does not infect the patient and acts only as a transport vesicle. In the bloodstream, AAV9 enables delivery of the gene past the blood-brain barrier and into the motor neuron nucleus, where it releases the new gene to form a self-complementary episome. An episome is a length of DNA that can exist independently from the patient's genome. Expression of the new gene is driven by a promoter, which results in continuous protein production in motor neurons. Overall, by replacing the mutated SMN gene, Zolgensma helps restore essential muscle function.
So what went into developing this drug? Well, it all started back in the mid-1960s when adeno-associated viruses were originally discovered using electron microscopy. The Burns laboratory developed ways of replicating the AAVs as well as their recombinants. The recombinant AAV2 was then developed to be used as a gene transfer mechanism in eukaryotic organisms, specifically for the treatment of cystic fibrosis in 1990. From there, it was theorized that the recombinants of AAVs could be made for a multitude of diseases by developing them to have a higher attraction for their target cells. With this, dozens of AAVs were discovered to target specific parts of the human body. In the mid-2000s, Brandon Casper was studying SMA and wondered how he could cross the blood-brain barrier and deliver treatment for this disease. He discovered some variants of AAVs were indeed crossing the blood-brain barrier as well as targeting muscle. Sometime later, AAV9, which targets the muscles and the central nervous system, was discovered. He used different aged mouse models with SMA to study the effect of AAV9 on the disorder. To his surprise, the treatment worked. This would lead to the creation of Zolgensma, clinical trials, and the FDA approval of the drug in 38 countries.
In order to measure the effectiveness of the drug as well as any potential side effects, various clinical trials and studies must be conducted. This stage of drug development is the most challenging and comprehensive step. The most well-known and successful clinical trial completed before the approval of Zolgensma is called the START clinical study. This clinical trial included 15 patients with SMA type 1. These patients were split into 2 groups. 12 were given a high dose of the drug and the other 3 were given a small dose. After a 24-month period, results showed that all of the patients who were given a high dose were able to breathe without support, and 75% of these patients could sit up without assistance for more than 30 seconds. On the other hand, all patients who received the low dose required breathing support with no improvement seen in muscle movement. Results from various other clinical trials suggest that Zolgensma is effective in treating spinal muscular atrophy by decreasing symptoms, reducing the need for permanent ventilation, and improving survival rates amongst patients.
When evaluating a new drug discovery, it is important to look for improvements that make it both clinically effective and cost-effective. Firstly, modifications to the vector could improve its gene transport stability. Recombinants of AAVs can be made to contain a new gene that exceeds its wild-type genome length by more than 100%. However, these vectors have been shown to grow poorly since the genome tends to be either unpackageable or unstable as a result of spontaneous rearranging. In comparison, vectors with genomes of approximately 105% of the wild-type length are relatively stable. Other modifications to the vector include modifications to its capsid, the protein shell of the virus, or addition of specific antibodies to the AAV. This would enhance the vector's specificity towards targeting motor neurons. Zolgensma is a one-time treatment that costs $3.7 million in Canada, making it the most expensive medication in the world. A reduction in the price for the drug would make it more accessible for families in need.
Overall, Zolgensma provides a cure for a deadly disease and offers hope for children with SMA, helping them grow stronger day by day.